specimen. So the first specimen in this phylum which we will study is the Cycan, or it is also known as Cypha. Okay, so these are, as I told you, that these specimens belong to phylum Poriferans, and Poriferans means the organisms bearing pores. Okay, so they have holes in their body which perform different functions. Just a minute. Pores in their body, and through these pores, various uh, processes occurs, like nutrition can occur, right? Release of water can occur, intake of water can occur. So all these things are performed through these pores. So the first specimen is the Cycan or Cypha, and you can see in the middle picture, this is the organism or the animal which is attached to the surface, to the bottom. Okay, so these are aquatic animals. Okay, so mostly they remain attached to a substratum. So that means they are grounded. They are not free to move. Okay, they can move, but they try to remain attached to a substratum or a surface. Right. So you can see also in case of this figure, that is the line diagram, that these are attached to a substratum with the base. So there is a base present, and with the help of this base, they get attached to the substratum. Then you can see the body of these sponges. They are also called as sponges. Okay, so poriferans, आप अगर book में पढ़ेंगे, they are also referred to as sponge or sponges. So we will discuss why they are called as sponges. So these sponges or poriferans, they have the cylindrical body. That means the cycons. And these cylindrical body can measure up to 4 to 5 centimeters. All right. And after reaching a particular length, they divide. Okay. They reproduce. And the reproduction can be of various types, both sexual and asexual type. And the most common form of reproduction in case of cycan is through budding. So these birds are formed, and these birds can generate into a new organism. All right. So a cylindrical body is present, and all over the body there are pores present. Right. So these all dotted structures are nothing but pores. All right. And on the apical side, that is on the top side, there is an opening which is called as osculum. समझ आ रहा है? So osculum are basically like the oral opening in case of other animals. So mouth जैसे होता है, same function is performed by the osculum. And then there are dermal ostia. So ostia basically are the holes which are present on the surface. Dermis जो होती है, epidermis जैसे हमारे केस में होती है, same there are the outer covering here. So on the covering over the body, main body, there are pores present. So these pores are not called as osculum, but they are called as the ostia. And because they are present over the dermal layer, they are called as the dermal ostia. Then there is a peculiarity in case of these poriferans, that the body is covered with spicules. All right. So spicules are basically the spines, or they are pointed structures which have varied shape. We will discuss that in today's class only, that they have a varied shape, and based on the shape, size, number, always remember that based on these spicules, on the number of the spicules, size and the shape of the spicules, these are classified. These organisms, that is poriferans, they are classified. So that is one of the major taxonomic characters. All right. In the lab, where you can see that these small holes are present. Okay, so if you look more closely, मैं इसको upload भी कर दूंगा Google Classroom पे, and you will be able to see a better view. in the enlarged picture, that holes are more clear in case of the preserved specimen. And can anyone tell me how these specimens are preserved, or what is the preservative used for preparing or preserving these animal specimens?
Formalin.
Yes,
Formalin.
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 Yes. So formaldehyde, basically an aldehyde, it is used to preserve the or the fix the basically the animals. And what is the concentration of that formaldehyde? It is not 100%. Rather, it is just 10%. So formalin is actually 10% volume by volume formaldehyde. So how will you prepare if I tell you to prepare a formalin solution for preserving the specimen? So you will take 100 ml of formalin or formaldehyde and add 900 ml of distilled water. Okay, and sometimes what uh, what the scientists do or the researchers do, instead of adding distilled water, they prepare formalin or formaldehyde in phosphate buffer or any other kind of buffer. So the simple reason for preparation of formalin or formaldehyde in buffer is that buffer is isotonic. All right, and because of the presence of those salts, these specimens could be preserved in much better way when we are using buffer as compared to the distilled water, because distilled water is hypotonic. तो क्या होगा, जो salts हैं इसके अंदर से they will move from inside to the outside environment, so they will shrink. समझ में आया? तो आपने hypertonic, isotonic और hypotonic सुना होगा वर्ड्स, and that we have to take care in case of preserving the specimens as well. So still, how much precaution you take, you preserve them in formaldehyde, and sometimes we also preserve them in 70% ethanol. ठीक है ना? तो 70% ethanol भी आपको मिल जाएगा कहीं कहीं बुक्स में. We preserve these specimens. So after taking all these care, still you cannot preserve them in the natural form. So after preserving, it looks like this. So in exam, we can show you a picture of the specimen in the jar or in a tube, all right? Or we can show the picture in the form of the natural habitat, all right? So you have to be able to identify that, okay, this is looking like this, but we will not show you a picture which we have not shown you earlier. Okay, so we will show you from all the pictures which we have discussed in the class, not from internet or any other source. So always try to remember that these specimens will be shown in your final exams as well. All right. So when you look at these structures here, that they have the growths which are present at a particular regions. So these are nothing but the budding process. So bud has formed and this has been reproducing asexually. All right. So this was about the first specimen, which is Cycan. Then comes another poriferan that is called as the Hyalonema. Can anybody tell me the name of this, common name of this uh, specimen? Hyalonema का common name क्या है? ग्लास रोप स्पंज का नाम सुना है? ग्लास रोप स्पंज नहीं सुना. So this is
No,
called
sir.
as the glass rope sponge. Why this is called as the glass rope sponge is that you can see here that the tail or the posterior part of the specimen, it looks like it is made up of glass. All right. So these are the spicules basically, which are siliceous. Silica का बना होता है. So glass is also nothing but silica. All right. So what happens in case of these sponges, that is hyalonema, that the posterior part, it resembles like a rope, all right, and they have the twisted fibers present there, and the body, main body, is spherical or oval, is rounded or oval in shape, and you can see the opening, which is again called as the osculum. Main body is rounded shape, all right, and the top opening is called as the osculum, like in case of the Cycan as well, and the body is covered with pores, which are called as the ostia. समझ आ गया? And then there are some symbiotic polyps present, okay, over these region. Okay, so these neck-like region, or you can say basically the tail-like region, they have the symbiotic polyps which are present there, and below that the siliceous spicules are present which are entangled. जैसे कि रोप होती है उसमें हम फाइबर्स को एंटेंगल करते हैं, उसी तरीके से same in case of hyalonema, the spicules are present which are siliceous in nature, and they get a root-like structure, and therefore it is called as the glass rope sponge. All right. Then comes the another major sponge which is euplectella. इसका तो नाम पता होगा सब, common name. euplectella का common name. मेडिकल की तैयारी नहीं चल रही है लग रहा है.
Venus flower basket.
Very good, Venus flower basket. Okay, so this is called as the Venus flower basket, and this animal, basically this is the shell of the animal. This was given as present in the weddings in Japan. Okay, so Oriental countries जो हैं वहाँ पर these beautiful structures were given as presents in weddings. Okay, so these siliceous structures are basically look like the glass fibers, and you can see here that at the posterior end there are the tufts of roots which are present. Okay, so these are the root like structures which have the fibrous tufts which are present, and on the main body or the over the cylindrical part there is a laceous work of the spicules. So just like the laces are there, the बेल होती है, उस तरीके का arrangement होता है इसके ऊपर पूरी body के ऊपर. And you can see here that there are some holes present. So this is the peculiarity of all the poriferans, that they have holes in their body, and these holes are not called as ostium or ostia, rather they are called as the parietal gaps. Okay, so these holes which are there, you can clearly see these holes are nothing but the parietal gaps. Why they are called as the parietal gaps? Because parietal refers to the term which is used for the body wall or covering of an hollow thing. If something is hollow and there is a covering of that hollow mass, then that is called as parietal. So in this region, that is the parietal region, which is the covering, hollow covering, there are the holes present, and they are called as the parietal gaps. Then there is a oscular sieve plate. So osculum जैसे वहाँ पर present था, इसमें एक plate होती है. So these you can see here, it is clearly visible that the top or the anterior part contains the oscular sieve plate, which helps in the ingestion. All right. Then we have to study the another specimen, which is the Potryon, and you can see it looks like a trophy, okay, or a pot. So from that the name has been derived, that it looks like a pot. All right, or it is also called as the Neptune's cup. Okay, so Neptune is basically the name of mythological Roman god, and that was having the habitat in water. So that was a water god in case of the Roman mythology. So Neptune के नाम पे इसको commonly we called it as the Neptune's cup. So the cup of the Roman god Neptune, right? And basically it is quite big in size, and it can measure in meters. So most of the Potryons, they measure about 1 or 1.5 meters, and some of them have been reported to be more than 2 meters. All right, so they are quite large in shape. Then, as you can see on the picture on the left hand side, there is a big opening there. So this opening is actually the ostium. Right? So osculum is present at the anterior end, which is quite large, and the skeleton is made up of siliceous monoaxons, and which are basically the spicules present. So you cannot see spicules with your naked eyes, but when you see this under the microscope, and more importantly under the electron microscope, so electron microscopic pictures have revealed that this quadrion is covered with spicules, which are the siliceous structures which are present over the body, and these spicules are present all over the body, and they have monoaxon. That means they have a single axon, यानी कि single axis उसके होती है, that they are present. We will discuss what are the different types of spicules, so don't worry about that. We will discuss. All right, and another thing which is important, that these are again sedentary because they are such a large size. They remain attached to a particular substratum. Right? So they remain attached to a bottom of the ocean or bottom of the sea. They are attached, and they have a cylindrical stalk. So they have cylindrical stalk basically means they have a narrow portion which is present at the base of the body, which helps them in the attachment to the substratum. All right. So that is the characteristics for quadrion. Then comes the bath sponge or euspongia, which is again a poriferan, and you can clearly see on the picture on the left hand side that this looks like a ball of sponge. Okay, so जो हम लोग sponge use करते हैं in kitchen as well as in bath, so they have similar structures like this. Okay, so it looks like जो आप अपना utensils को wash करने के लिए, clean करने के लिए sponge use करते हैं, it looks similar to that. Okay, so when it is extracted or when it is taken out from the sea, it is actually marine. So always remember that euspongia are marine poriferans. So they will not be present in the fresh water. So you will only find them in seas, okay, in oceans, and not in the rivers and ponds. Right? So these euspongia basically they are the bath sponge, and they have these pores which are extensively present all over their body. Okay, and you can see the size of the pores which are more prominent in the picture on the left hand side, which is a sponge which was extracted, which was taken out from the sea, and these pores are basically the oscula. Osculum वर्ड ये नहीं यूज करेंगे जो कि हमने पहले यूज किया था. Osculum means singular. All right. So plural, when more than one oral opening is there, that is called as oscula. So there are numbers of oscula which are present. तो आप जब diagram बनाएंगे तो आप उसमें 3-4 को आप लेवल करेंगे कि they are the oscula, and try to make them a little bigger. All right. And this is the preserved specimen of the same euspongia, and again they are attached to the substratum, which helps in their staying at a particular place. All right. And again these sponges, when you look under the electron microscope, you will find that these have the spicules which are present over them. All right. And these spicules are, no, not in case of the euspongia, I'm sorry. In case of the euspongia, these spicules are not present. All right. So that is an anomaly for the absence of spicules. They are not present over the euspongia. And earlier in the ancient times, they were also used for taking baths. Okay, so they were just like used as these sponges. So people used to take them out, kill them, and then they used to bleach, and finally they used to use them as a sponge, in taking bath. Alright, so they have enormous capacity to retain water because of the presence of all these pores. Alright. Then comes the spicules, which I was talking about. That when you look at the body itself, you will see that these are basically the smooth structures. Okay, in this case also, hyalonema, and in case of cycle. So apparently they look like if their body is smooth, but when you observe these poriferans, that means the surface of the poriferans, they are covered with a number of variety of spicules. Okay, so what are the spicules? Basically, these spicules are the uh, fine structures which are present on the skeleton or the body of the sponges. Okay, and they are sclerotized. Okay, and uh, these spicules are siliceous in nature. So because of that, they give an appearance of uh, glass-like nature when you look under the microscope. So based on the axis, okay, these spicules are divided into a number of types. The first one is the monaxons or the monoaxons. तो monoaxons को हम monaxons लिखते हैं. So that means only single axis is there, a single plane is there, which is present, and that is called as the monoaxon spicules. And monoaxon spicules are present in case of this one, that is portion. Always remember. Portion के केस में monoaxon spicules present होंगे. In others, majorly triaxons, tetraxons, and polyaxons spicules are present. So triaxons means they have three axes. The first is this axis, second is this axis, and third is this axis. So it is not like these have 6 rays, but 3 axes. So don't confuse that this is polyaxon. No, this is triaxon, because the axis is from this point to this point. This is one axis. Right? So x-axis जैसे होती है तो नेगेटिव एक्सिस और पॉजिटिव एक्सिस. तो पॉजिटिव वाई एक्सिस आप मान लीजिए इसको, और ये नेगेटिव वाई एक्सिस, बट स्टिल इट विल बी ओनली वाई एक्सिस. इसे z एक्सिस मान लीजिए, इसे x एक्सिस मान लीजिए. x, y और z, तो 3 एक्सिस हो गई. दैट्स व्हाई इट इज कॉल्ड एज द ट्राइएक्सॉन. Triaxon, 3 axis. Tetraxon, 4 axis. Right. So 4 axis can either be in terms of the elongation at one side only. Okay. So इसमें क्या था? Bidirectional movement था spines का, bidirectional localization था. But in this case, it is on the first figure which I've taken here. They are present only on one side. So they are 4 in number. So they are called as the tetraxons. Alright. Another example of tetraxon is on the right side. This is also a tetraxon, and this is called as the amphidisc. Okay, so this is the amphi means both on the 2 sides. So at both the sides, the disc is present, the disc-like structure is present, which have again 8 rays and 4 axes. So इसके आप opposite side देखेंगे, there will be another ray, so that will be one axis, second axis, third axis, fourth axis. So these are again tetraxons which are there. Okay. Then comes polyaxons. Poly means many. So a number of axes are there, that is more than 4. It is called as the polyaxon. So polyaxon, you can see clearly there are a number of axes which are present, more than 4. Then comes the desma, which is basically either can be monoaxon or triaxon or tetraxon, and they keep on depositing the silica over them. Okay, so they become siliceous or they become more dense by the deposition of successive layers of silica. Okay, on either monoaxon, triaxon, or tetraxon. All right. So this is another form of spicules. Then the final form of spicules in case of the poriferans is the spherical type. You can see they look like spherical balls and they have these siliceous pointed structures which are the spicules present. Right. So these basically six major types of spicules are there in case of the poriferans, and these helps in the identification of the specimen. Okay, so if you look at the specimen by the naked eye, you will see like this or like this or like this. But when observed under the microscope, you will find that these have been covered with the spicules or they are siliceous in nature, and because of that, and because of their size, their number, and their shapes, they can be used as a taxonomic character. So this is the line diagram for the spicules. So you can see monoaxon, just like this one, which is tapered at both sides. That is called as oxia. It is an example of monoaxon spicule or monaxon. Then the triaxon, you can see here that three axes are there. The line diagram for triaxon is this one. First one, you draw only this one or this one, that there are only three axes. Okay. Then for the tetra, I have told you about the amphidisc and this one, that is tetracytine. Okay, so in this tetracytine, there are four axes which are present, and in case of amphidisc, four axes are there, but they are present at both the ends. So that is why it is called as the amphidisc. The disc is present at both the ends. And this one is the desma, which is actually a spicule where the deposition of silica is taking place again and again, and that's why it has taken a more robust shape. Okay. Spherical का इसमें मैंने diagram इसमें book में था नहीं, तो I have not taken, but you can simply draw the spherical one that make a spherical structure and make pointed structures which are the spicules. Right. Then the final specimen which we have to study in case of the poriferans is basically the gemmules. Okay, so gemmules are nothing but the asexual reproductive structures which are ball-like. So you can see these yellowish structures which are oval in shape, and these are nothing but the gemmules of spongilla. And spongilla, unlike euspongilla, is a freshwater sponge or freshwater poriferan. So it will be found in ponds, it will be found in lakes, it can be found in rivers. So wherever there is fresh water, it can be found easily. But it will not be found in case of the marine environment. In oceans or sea may, you will not find these. All right. And when you look at these under the microscope, these each globular structures or ball-like structure, then you will find that it is made up of two membranes. So first is the outer membrane, which is present, that covers the gemmule, and then there is another membrane, which is the inner membrane. Okay, and again, both in the inner and the outer membrane, there are the spicules present, and because they are having one axis, they are called as the monoaxon spicules. So in case of spongilla, that is the freshwater sponge, you will find that monoaxons are present in the gemmules, and there are two layers, that is the outer layer and the inner layer. Then another important thing about the gemmule structure is the presence of archaeocytes. Okay, so archaeocytes are basically the specialized cells which are totipotent in nature. तो आपने वर्ड पढ़े होंगे totipotent, pluripotent, multipotent. So these stem cells are basically totipotent. That means they can differentiate into any type of cell. All right. So these archaeocytes, these are the totipotent cells which are like the stem cells, not actually the stem cells, but they behave like the stem cells as they have rich cytoplasmic content and etc. So they can divide upon the requirement into any type of cell. So whatever be the requirement, they can divide into that type of cell. So suppose if they want to replace a particular part of the body, they can be transformed into that particular cells which have to be replaced. Or if they want to be replaced into the choanocytes, so which are important cells which are used in the nutritional process. So choanocytes may replace हो सकते हैं. They can be replaced into the reproductive structures. So here what we are seeing is a gemule of spongilla, which is an asexual structure. And always remember that under the normal conditions, gemule formation is not occurring. Okay, so this only occurs, that is gemule formation only occurs when there is unfavorable condition. और हमने पढ़ा था जैसे अमीबा के केस में अनफेवरेबल कंडीशन में क्या होता है, सिस्ट फॉर्मेशन हो जाता है एंड इट स्टार्ट रिप्रोड्यूसिंग बाय मल्टीपल फिजन. So therefore, likewise, in case of the poriferans also, these poriferans, especially the spongilla, which is the freshwater sponge, they make gemmules, alright, and these gemmules, they contain an important cells or cluster of cells which are the archaeocytes, and these archaeocytes are the totipotent cells which can transform into any type of cells upon the coming of the favorable condition. So when the conditions become favorable, these archaeocytes, they can come out of the micropyle. So micropyle आपने word seeds के केस में पढ़ा है ना, seeds के केस में micropyle botany में पढ़ते हैं.
Yes, sir.
So like in case of the seeds which are present, the micropyle is the opening through which these archaeocytes can come out and they can develop into the particular type of cells. Okay, or they can give rise to another sponge. Okay, so they can be used as a source of vegetative reproduction or asexual reproduction.
फेवरेबल कंडीशन ये होगा सर,
जी,
फेवरेबल कंडीशन पे मतलब कि चेंज होंगी.
unfavorable condition means because they are present in the freshwater region, the sometimes water level goes down, okay, or the temperature rises too much. So those are the conditions which are unfavorable. So upon those conditions, these sponge, in order to protect their progeny, they have to reproduce. Okay, so they start producing these gemmules, and when the conditions become favorable, that means there is plenty of water, temperature is low, harsh conditions are not present, these archaeocytes, they can come out of the micropyle and start developing into either the whole organism or a specialized cell, whatever is the requirement, because they are the totipotent cells. Always remember that. Okay, so this was all about the specimens which you have to study. So in total there are, one is the Cycan, second Hyalurone, Euplectella third, Fortreon fourth, Euspongia 5, Specules 6 और ये 7 हो गया gemule. So these are the 7 specimens which you have to draw in your notebook, and as I have told you earlier also, that you have to draw only the line diagrams. You don't have to paste these pictures, you don't have to draw these pictures of the actual specimen, you have to only draw the line diagrams and write the comments and the classification. Alright, so book आपके पास है, उसी में से आपको डिटेल मिल जाएगी. You write about these, right, and then submit. I will create the link. और लास्ट लेक्चर की